Hi, welcome to General Biology, Chapter 1, your lecture notes scheduled on your syllabus for Wednesday, August 19th. Our objectives for Chapter 1 include an overall understanding of the concepts of general science with an emphasis on life science. We will cover the methods of conducting an experiment, referred to as the scientific method, and get an understanding of the hierarchy involved in organizing the massive amounts of data accumulated through observation of the world around us. All life is founded in chemistry, which takes us down to the atoms that form molecules. No life exists without water, and we will discuss the molecular properties of water that makes it so special and unique to this planet. As far as we know, it's the only planet that is covered in water in our solar system. Biology is defined as the scientific study of all living organisms, and this includes the study of their interactions with one another and with their environment. As we progress through Unit 1, we will talk about the macromolecules that the cow is eating in this picture and the gas exchange with the environment, as well as how the oxygen he breathes in with every breath drives the conversion of food into a more usable form of energy that will be made available to the cells which make up the tissues, and those tissues form organs that work in the organ system to support the animal. Our workhorse in biology is the microscope. This piece of equipment allows us to make observations and apply the scientific method to organisms that are not always visible to the naked eye. We will use logic and a specific process when it comes to interpreting data collected during our observations of the natural world around us. Please try to commit to memory the following examples I've given to you so that you may recognize the difference between inductive and deductive reasoning. Inductive reasoning is a process where we record our observations, like in a lab notebook, and then we draw conclusions from the data. This process takes you from the specific to the more generalized based on patterns in the data. Scientists use this method to form hypotheses and theories. Scientific results can generate two types of data, either quantitative, where you have numbers assigned to different categories, like what is shown here on the graph to the right. Another perfectly reasonable type of data we see often in the lab are qualitative results, here on the left. In this case, we see a color change that indicates a positive reaction when compared to a negative control. This is an example of the Benedict's test, and we will indicate the presence of sugars in the sample by a color change. The bright orange is positive, and the royal blue is actually negative. So you don't generate numbers, but again, the color change is another valid form of data that we often use in the laboratory. Deductive reasoning starts out with a general statement or hypothesis and then examines the possibilities, which is called forecasting, in order to reach a specific logical conclusion. We are then able to predict what the observations should be if the theory were correct. We go from the general, the theory, to the specific. Here we see a male polar bear that is approximately 1,000 pounds underweight. Why? Because of climate change. Do you see any snow in this photograph published in National Geographic from 2 summers ago? No, there is none. Therefore, we conclude that the bear can no longer hunt for food in his environment due to these environmental changes. For in-class participation points, email me your answer to this Granny Smith apple question. What type of reasoning do we use when we see a green apple and say that it is a fruit, when you know that all apples are fruits? What do we mean by science has a two-pronged approach? This is the uh, property that we recognize science as generating scientific evidence using specific predictable processes called the scientific method. Science is also defined as a body of knowledge. When enough data has been generated, a theory can be developed based on a set of observations supported by facts. A theory is a widely accepted general statement about a fundamental concept. Most of the time, scientists work using deductive reasoning. They have a specific question, usually based on an observation in nature, and they want to test it to see if their working hypothesis is correct or not. Here, we compare the major difference between a hypothesis and a theory. Your working hypothesis is your proposal for an explanation based on your observations. This is a testable process through experimentation. A theory is much broader and more general than your hypothesis and can explain a great many observations. A theory is supported by a large, growing body of evidence that has not yet been falsified. So we use the scientific method. It's something you should become very familiar with and be able to apply to any and all work done in the laboratory setting. This is a series of steps that all scientists use repeatedly whenever they're working. First, you make an observation here at the top, and then that leads you to a question. And this question leads you to the formation of your working hypothesis. Once you have your hypothesis or your idea, you'll need to test it through experimentation. From your careful experiments, you'll collect scientific results down here at the bottom. From those results, you'll draw conclusions. Sometimes your conclusions are unexpected and may lead to further questions. And so this method cycles again and again. We often rely on our colleagues and peers to review our work. Collaborations can be formed this way, and some work will get published. Experimental design is a very important aspect of experimentation if we are to trust our results. All experiments will have an independent variable, which is part of the experiment that you control or you as the researcher can manipulate. The dependent variable is usually what we measure. This is where your units assigned to any numbers generated in the experiment are very important. For example, did you measure in inches or centimeters? And all tests must have a control. This will be your standard by which all other data will be compared. Maybe you want to have a blank or a zero in order to accurately determine change. Your experiments will not be valid unless you run controls during the test conditions. Positive controls show change, and negative controls show a lack of response. These are needed in order to determine the amount of change taking place in your variables, which are your test groups. Using logic and the scientific method, biologists must often consider what is alive? What are the criteria for determining if something is alive? What are the collective properties that make something alive? And how do we find organizational structures and then place them into some sort of hierarchy? Are the rocks and the air and the water shown in this photo alive? What we look for in this list, it's in this list and may seem obvious to you, but let's go through them. If we're going to qualify something as being alive, we will find organization and visible structural order. Anything that's alive is going to be capable of growth and development, reproduction. It's going to use energy, and all life as we know it, has a cellular basis. Anything that's alive is going to be able to respond to stimulus, and it's going to be capable of what's called homeostasis. That's a process of regulating itself under different environmental circumstances. You can pick on your internal temperature. Okay, we tend to be 98.6 degrees Fahrenheit, and our body regulates our temperature whether we go outside in 100-degree weather or we're outside in the freezing cold. Our body temperature usually stays about the same. That's a great example of homeostasis. And then all living entities are capable of adaptation and evolution over time. The hierarchical organization of life here on Earth starts at the atomic level, down here at the bottom. We're going to start small and work our way up. We will study atomic structure in more depth in Chapter 2. The next step in complexity is the molecule. In this photo here at the bottom, we have a molecule of DNA. Certain organelles contain DNA, and the cells that contain organelles have a special name. These are eukaryotes. So at this level, the cell has a nucleus, and you can see that dark staining, centrally located little dot in the middle. That would contain the DNA of a eukaryotic cell. Cells that have common function can form tissues, and there are 4 primary tissues that form the organs. Organs can work within a system of different organs in order to support the whole organism. And a microscope is required to view the cellular level and smaller. Here's a look at the same concept using a plant instead of an animal. And yes, the leaf of a tree is considered an organ. It's made of tissues that are working together, and the organ is capable of gas exchange and fluid exchange. You can see the veining in the leaf. So this is an organ. You can go backwards from the organ to the tissue, to the cell, to the organelle, to the molecule, and to the atom. You can go ahead and look at figure 116 and use the visual connections on page 24 in your ebook. Okay, so to complete our look at Chapter 1, the video here on this slide, make sure that you watch this video on hierarchy, and by now you should have already viewed the video contained in the announcements covered on Monday, and that talks about the Earth and stromatolites, which contribute to the Earth's current atmosphere. Biodiversity is all around us, from the tiny prokaryotic bacteria to the king of the jungle. Every living entity on Earth has a role to play and fits into a web of life that is interdependent on each other. So we conclude our tour through Chapter 1 with the Tree of Life. We talk about this in more detail. We'll talk about phyla and the 3-domain system of categorizing all life on Earth as we progress through this semester. Thank you for listening and taking notes during this video. Email me if you have any questions about the material that we just covered.